Okay guys, going on to lecture 2, which is going to be an introduction to our sensory receptors, like touch on our skin, hearing, sight. It's really exciting. So strap in and pay attention. Right now, I'm wearing a University of Southern Mississippi sweatshirt. It is pale yellow with black writing, and it's ribbed. I'm also wearing gray sweatpants. I'm going to ask you about what I was wearing to make sure you're paying attention. That's why I said that. So, on to slide 1. We're going to be talking about their general senses and very specific senses. General senses can be felt in many areas. That includes touch, pain, pressure, vibration, stretch, temperature, the awareness of your body position. And then there's the specific senses, which are perceived only by specific localized receptors, like sight, smell, taste, hearing, and equilibrium. So for general senses, they can be felt like across your entire body, like all throughout, anywhere you have skin, from the top of your head to the bottom of your toes, whereas in the specific senses, there's literal, sometimes organs dedicated to that one thing. Like you have eyes for sight, you can't see out of anywhere else. So when we're going to be talking about receptors, what is a receptor? It is a receiving unit of the body that responds to a stimulus. Or stimuli for plural. Stimuli is just environmental input. For example, whenever you see something, the light that is entering your eye is the stimulus. It is sensitive to specific modalities and can be classified according to them. And modality is just a fancy way of saying a type of, so like, that's like having eyes specifically for seeing. That's its modality, is sight. And ears and the inner ear, all that kind of stuff, has a hearing modality. Excuse me. Then there are classifications. So specific modalities, like photoreceptors, which detect light. Thermoreceptors detect temperature. Proprioceptors detect changes in tension. Nociceptors detect pain. Baroreceptors detect changes in pressure, like barometer for the weather. Chemoreceptors detect changes in chemical environments. Right here is just a fun little slide of the different general sensing areas in your skin. For touch, so we have the Merkel's discs up top. There are free nerve endings all throughout the skin. Ruffini corpuscles, root hair plexus on that purple thing, and the Pacinian corpuscles, which are these, in this case, green sort of orb-like structures with nerves on the inside. So moving on to taste and smell specifically. We have gustation and olfaction, which is exactly what I just said. Gustation is taste, olfaction is smell. With gustation, it uses chemoreception on your tongue. It is received in the tongue, the soft palate, and the pharynx, all the way through the beginnings of your throat. The nervous system anatomy you want to recall here. The sense of taste is transmitted by facial, glossopharyngeal, and vagus nerves. So we went over those nerves in the last lecture. These actually pick up the chemoreceptions on your tongue and interpret them as taste. They are interpreted in the post-central gyrus of the parietal lobe in the brain, which we also talked about last time. And olfaction with smell is another chemoreception. It's received in the olfactory epithelium, which is lining the nose on the inside. The nervous system anatomy to recall here is that the sense of the smell is transmitted by the olfactory nerve and is interpreted in the temporal lobe. So these nerve names should be easy to recall with these specific functions, because glossopharyngeal just literally means like having to do with your mouth and your throat. So it makes sense that it's involved with gustation and the transfer of those chemoreceptions. And for olfactions, it's just the olfactory nerve. So when you really think about it, it's not too difficult to memorize. Moving on to your eyes and vision. So it's important to know that all eye models our right eyes. I am actually not sure why that is. I think it's just for consistency's sake, because the nerves would be flipped in the other eye. So here are the different points that I have mapped out. We have the sclera, the choroid, the retina, and the optic nerve. And I have small definitions. The sclera is the white of the eye. It's a fibrous outer layer. The choroid is nutritive, vascular middle layer. It's got a lot of blood vessels in it. The retina is a tri-layered neural and photoreceptive innermost layer. And the optic nerve is where the axons of ganglion cells exit the eye. So the sclera and the choroid are protective, they cover the eye. The retina actually tries to gather this information from the light entering the eye, and the optic nerve transmits that information to your brain so you can perceive it as light. So the conjunctiva is not visible on this model. You don't really need to know it anyway, so I'm not going to go over it. We've got some muscles going on. The eye muscles, we have your superior rectus, the inferior rectus, the medial rectus, the lateral rectus, the superior oblique, and the inferior oblique. These should not be difficult to memorize either, because you really only have to memorize one word and just the direction. And if you already know the directional terms from the first lecture, you would be able to guess what these were. That would be good for our practical. So here's some extra terminology for the words that you may not already understand. Oblique means it pulls to the side, rotates laterally or medially. Rectus is just a word to say it pulls straight back and up or down. And medial pulls inward while lateral pulls outward. And this is how you're able to move your eyes, is by just, using these muscles to contract. Some basic stuff you already know. The cornea is that glassy covering on the front. The iris is where the color of your eye is. The pupil is the hole. It is not an actual structure. It is just a hole that the iris expands or contracts in order to focus the light onto your lens, which is in the back of your eye, which flips the image and is then interpreted. And so you can see it says superficial on this side. That's because it's the front of the eye, and deep over here with the lens because it's way inside the eye. Cavities and chambers. The eye is divided into the anterior and posterior cavities. The anterior cavity is filled with aqueous humor. The posterior cavity is filled with vitreous humor. The anterior cavity further divides into the anterior and posterior chambers. The anterior chamber extends from the cornea to the pupil/iris, and the posterior chamber extends from the pupil/iris to the lens. So if you can see, these are just the terms about the different portions of the eye. There is a lot to go through. So when we're talking about the posterior cavity of the right eye, lateral, anterior, internal, and posterior internal view. Yep. You get a clearer look with this model of the vitreous humor, the ora serrata, the macula lutea, the fovea centralis, and the optic disc. Oh dear, that's in the way. The ciliary body is hidden down here, but that's just what it says, ciliary body. Some more fun words. Talking about the lacrimal sac. That is your tear duct, where your tears are stored. The optic chiasma, and the lacrimal gland, which is that big brain-looking structure. It is covered on my screen. I don't know if it is on your screen, actually. You're probably confused about what I'm talking about. Then there's just a better view of the muscles that we talked about earlier that actually move your eyes. You can see they're a lot longer than it may have been perceived at first. They pull all the way back. Okay. So this is the pathway of light through the eye. It enters through the cornea, through your iris, where it is focused by the pupil, passes through the crystalline lens, goes all the way back to reflect on your retina on the focal point where the optic nerves then receive that message. Moving onward to your ears, hearing, and balance, which they are connected. So you're familiar with ears. Sound waves enter our ears through the outer ear, they go in through the middle ear, and they are then interpreted in the inner ear. This is a better look at the structure if you were to cut someone's head open. Thank goodness for models, am I right? I'll assume you laughed. Some more specific names. Your auricle is your ear. You got the top of your ear, the helix, which is where it starts to fold, and the lobule, the lobe of your ear, on the outer ear. The middle ear, we have the external auditory canal, which is where you shove Q-tips. And then the tympanic membrane is your eardrum. And it hurts if you burst it, so don't do that on purpose. Moving into your inner ear, you have the different ear bones, the temporal bone, and the auditory tube. So with these very small little ear bones, you know, the incus, the malleus, yeah? Yeah, excuse me. Then moving on, we have the vestibular cochlear nerve, the cochlea itself, the semicircular canals, the ampulla, the vestibular cochlear nerve, semicircular canals, cochlea, and ampulla just focused on really hard. So you see it in the context of the actual head up in the top left, and then in the bottom right, you can just see it all pulled out. Here's another good window looking. We got an oval window, which was, I think, obscured in one of the last slides, and I forgot to mention it. We got our semicircular canals, stapes. There's the incus and the malleus and the tympanic membrane. Yep. So what goes on is that the sound waves actually vibrate these bones, which are then sort of amplified through your cochlea, and the vestibulocochlear nerve is really what interprets the sound waves. It brings it to the brain. So it's all just a pathway. Speaking of, I knew I was speaking too fast. So, equilibrium. Awareness of the head position in both stationary and moving phases are defined as equilibrium. Static equilibrium involves sensing the head position relative to gravity and is detected by vestibular receptors. Dynamic equilibrium involves rotational movement of the head and is detected by semicircular canals receptors. Utricle and saccule are the chambers in vestibules that each have maculae that have hair cells. These together provide information about the head position. There's a good image for you here on the right, where you can see sort of the interior view of the membranous labyrinth. And that's all for today, guys.